Welcome back to my channel Knowledge Chronicle.
Have you ever wondered how investigators catch criminals with just a drop of blood? Or how paternity is confirmed using just a strand of hair? How do scientists identify you even if you've never met them before? The answer lies in a groundbreaking technique: DNA fingerprinting. It's not science fiction, it's pure science. And today on Knowledge Chronicle, we're diving deep into this mind-blowing technology that makes every human truly unique. So stay with me as we unravel the real power hidden in your DNA. Let's start with a simple but stunning fact. 99.9% of the DNA in every human is exactly the same. Yes, we're all 99.9% identical in our DNA sequence. But that tiny 0.1% difference makes us all look, behave, and function differently. Imagine, if the human genome has around 3 billion base pairs, then 0.1% variation means around 3 million base pairs are different between any two people. That's 3 million unique letters in your genetic code, and those letters make you, well, you. But sequencing an entire 3 billion base pair DNA to compare people, that's exhausting and expensive. So what's the solution? DNA fingerprinting. DNA fingerprinting is a technique used to identify individuals by analyzing specific regions in the DNA that are highly variable. These are called repetitive DNA sequences. Imagine you and your friend both have a book. The main content is 99.9% the same, but in some chapters, there's a line that repeats, hello, hello, hello. In your friend's book, it might repeat 10 times. In yours, maybe 15. These repeated lines don't change the story, but they make each book unique. That's how repetitive DNA works. These repeating sequences are found in non-coding regions of the genome. They don't make proteins, but they occupy a large portion of your DNA. We call these repetitive DNAs, and when we isolate them using density gradient centrifugation, they appear as satellite DNA. Tiny peaks separate from the main bulk DNA. Now, these satellite DNAs are classified based on base composition. Are they rich in AT or GC? Length of segment, number of repeats. The two major types are mini-satellites, longer repeats like 10 to 60 base pairs found in clusters. Microsatellites, shorter repeats 2 to 6 base pairs repeated many times. These sequences are highly polymorphic, meaning they vary greatly among individuals, making them perfect for fingerprinting. Let's simplify a crucial term: polymorphism. Polymorphism means genetic variation that's inheritable and exists in more than 1% of the population. These variations are caused by mutations, and they happen in the non-coding regions more often because they don't directly affect protein formation or survival. These small changes, like a single base change or a long repetition, accumulate over generations and make every individual's DNA fingerprint different, even among family members. And yes, these differences are inherited, which is why DNA fingerprinting is a key tool in paternity testing and gene mapping. DNA fingerprinting as a technique was first developed by Sir Alec Jeffreys in 1985. He used a special kind of satellite DNA known as variable number of tandem repeats, or VNTRs. These VNTRs are part of mini-satellite DNA and are highly polymorphic. That means the number of repeat units varies so much that every person has a different pattern. Jeffreys used a southern blotting technique combined with a radio-labeled VNTR probe to visualize these patterns, and boom, a genetic barcode unique to every individual, just like a fingerprint. Here's how traditional DNA fingerprinting works. First, DNA isolation. You extract DNA from a sample: blood, hair, saliva, bone, you name it. Next, digestion. You cut the DNA using restriction enzymes. Then electrophoresis. You separate DNA fragments based on size using a gel. After that, blotting, you transfer these fragments onto a nylon or nitrocellulose membrane. Following this, hybridization. You use a radioactive VNTR probe that binds to repetitive sequences. Finally, autoradiography. You expose the membrane to an X-ray film to see the band pattern. Each individual gives a unique pattern of bands, like a barcode. Identical twins will have the same pattern, but no one else will. Earlier, DNA fingerprinting needed a lot of DNA, but now we have PCR polymerase chain reaction. This revolutionary method allows us to make millions of copies of DNA from a single cell. Yes, even one drop of blood or a single skin cell is enough to get a full DNA fingerprint. This has made DNA fingerprinting faster, cheaper, and more powerful. So where do we use DNA fingerprinting? Almost everywhere. Forensic science, crime scene investigations. Even a drop of blood or saliva can catch a criminal. Paternity testing confirms biological relationships with unmatched accuracy. Identifying disaster victims helps recognize remains when other identification fails. Wildlife conservation identifies and tracks animal species and genetic diversity. Medical research studies genetic diseases and population genetics. And remember, except for identical twins, no two people have the same DNA fingerprint, making it the most reliable identification tool in biology. So next time you hear about solving crimes using DNA or confirming a baby's parents, it's all thanks to the science of DNA fingerprinting. It's powerful, it's accurate, and it's changing the way we look at identity, relationships, and justice. If you found this video helpful and interesting, don't forget, your one like, share, and subscribe will help us create more valuable content. See you in the next video of Knowledge Chronicle, where knowledge meets curiosity.